to our next speaker, Daniel Gonzalez from the Kawasaki Institute of Industrial Promotion.
No. Thank you very much. First, I would like to thank the organizers to give us the opportunity to present the work we are carrying out at the Innovation Center of NanoMedicine in Japan to develop cell-targeted and brain-penetrating polymeric nanomicelles to treat neurodegenerative diseases. So we heard the blood-brain barrier is a big obstacle to therapies entering the brain. Therefore, strategies need to be developed to actively deliver therapies across the BBB. Despite its high impermeability, specific and active transport mechanisms do exist, such as the glucose transport protein, GLUT1. Therefore, nanovehicles may be created to target this transporter and deliver therapies into the brain. With that in mind, we have created glucose-decorated nanomicelles to be able to target the GLUT1 transporter. We have created these by mixing block copolymers consisting of an anionic polymer attached to a PEG chain with or without glucose termination. And we mix it with a cationic polymer, fluorescently labeled, attached to a PEG chain. This results in the self-assembly of a nanomicelle for about 30 nanometers with a polyionic core, fluorescently labeled, and a PEG shell with different percentage of glucose decoration. The micellar structure is then stabilized by EDC cross-linking to allow injection into biological fluids. And we have shown these nanomicelles enter the brain under specific glycemic conditions. So you can see that in free-feeding control mice, represented by the open circles, there is very little internalization into the brain up to an hour and a half. However, if the animals are starved overnight to lower blood glucose to induce translocation of the GLUT transporter onto the luminal side of the BBB, systemic micelle injection followed by injection of a high glucose dose leads to induce recycling of the GLUT1 transporter, leads to robust penetration into the brain, specifically of the glucose-decorated micelles, and specifically the 25% decoration glucose micelles. The 10% and 50%, there is still internalization, but it's not as strong. And importantly, this internalization increase is specific for the brain. So by fluorescence, we showed that there is no increase in penetration across all the organs that we tested, and it's specific for the brain. Following the in vivo work, we wanted to characterize further the internalization of the micelles by the endothelial cells. And to this end, we created in vitro BBB models by extracting primary endothelial cells from rat brains, and we treated them with either null micelles or 25% decorated glucose micelles. To model the free-feeding condition, we treated the cells with micelles directly in cell culture medium. And to do the glycemic condition, we first starved the cells of glucose by PBS incubation for 1 hour. Then the micelles were administered in a high glucose concentration. We plated the cells on glass cover slips to be able to visualize internalization by confocal microscopy, or on porous transwell systems to measure the actual transport across the BBB. So here we see the monolayer visualized by DAPI staining. And you can see that over the 24 hours, despite the high migration rate, it is non-random and it follows a very specific path. And more importantly, the endothelial monolayer integrity is maintained by, as you can see, by very little cellular overlap and minimal intercellular gaps. And importantly, the monolayer adopts a BBB phenotype as represented by intercellular occludin and Z01 expression and low paracellular transport. This visualization protocol was then applied to cells treated with the null micelles or with glucose-decorated micelles. And you can see that in the control condition, there is very little internalization of the micelles, represented by these pink aggregates. However, under the glycemic conditions, you see this robust increase in cellular internalization, specifically for the glucose-decorated micelles. There is slight internalization of null micelles, but it's not as marked. And we can see that by fluorescent quantification that the internalization is relatively stable over the 24 hours and no internalization for the free-feeding conditions. We wanted to check whether this internalization was actually translating into transport. And so we repeated experiments in the transwell assays, and we normalized against dextran and paracellular transport, and you can see that it is only the 25% glucose micelles under the glycemic conditions that have the increased transport across the BBB. Okay, then next, I have to hurry up. We wanted to see whether we could functionalize these micelles with proteins to be able to act as therapeutic cargoes or cell-targeting ligands. And to this end, we introduced an azide-terminated polymer to be able to bind DBCO-tagged proteins. And the therapeutic cargo that we were interested in was this anti-inflammatory protein CD200, because under normal conditions, this protein helps to maintain microglia under quiescent state. Therefore, if we promote brain entry, we would be able to revert the inflamed microglia to their resting state and induce neuroprotection. So just quickly, we bound DBCO to this protein following reduction by a malamide DBCO linker, and then we attached it onto the azide-containing nanomicelles. And we can see that by DLS characterization, there is increase in size, which importantly still retains a low PDI. And by fluorescent correlation spectroscopy, we can see that the protein is actually binding onto the micelles, as you can see by the increase in diffusion time between the free protein and the micellar protein. We treated brain macrophages with LPS to try to see if we could reduce inflammation by these micelles. And we can see that 24 hours after treatment, production of nitric oxide and reactive oxygen species is not affected by the naked micelles, but is significantly reduced by the CD200 functionalized micelles. So we show that indeed these protein functionalized micelles retain the ability to reduce inflammation. We next wanted to do cell targeting, and so we chose anti-excitatory amino acid transporter 2 antibody conjugated to FITC, and we digested this to fragment it to the Fab fragment, and we bound this to the micelles. And again, you can see that there is this increase in size with still retention of low PDI and a corroboration of protein binding onto the micelle by fluorescence correlation spectroscopy, and by the counts per molecule, we are calculating there are about 1.2 antibodies per micelle. Then we treated primary astrocytes or brain macrophages with the EA2 antibody micelle to see if we could target it to the astrocytes. And we can see that there is specifically increased binding of the micelles, of the Fab micelles, to the astrocytes, and as a negative control, we used macrophages. And the cell number is relatively similar, therefore we can conclude that there is increased targeting onto the astrocytes. So in conclusion, we've shown that are able to actively penetrate the blood-brain barrier. We've shown that these micelles can reduce inflammation, and we've shown that we are able to target the micelles to a specific cell type. Unfortunately, it's only the astrocytes at the moment. We are working on trying to actively target the microglia. And in the future, we want to combine all these strategies to be able to create a potent and specific nanomicelle to be able to treat neurodegenerative diseases. And with that, I would like to thank all my collaborators at ICOM, and specifically Dr. Anrako and Professor Kataoka, and the Japanese funders. Thank you.
Thank you very much for the interesting talk. Any other questions?
Mm-hmm.
So I'm interested in your glucose deficiency level.
Mm-hmm.
If it is achievable in clinical situation, because we all know that the brain glucose uptake, actually not that micelle, glucose uptake is very stable in fasting and also after postprandial state.
Mm-hmm.
So we think that it's insulin independent, so GLUT1. However, the glucose level is influencing what I mean is just the GLUT1 is increasing when glucose is deficient and decreasing in glucose is oversaturated. So back to the question, is it achievable level, or it is going to be a very low hypoglycemia so that you can so let your glucose-labeled micelle is going to taken up by then it is useless.
Yes, that's a very good point. Thank you for your question. We are worried about the level of hypoglycemia that we have to induce in patients, and especially if the patient is diabetic, it would be a severe problem. But I think if we can control the level of glucose specifically to, over time, allow enough accumulation of the glucose transporter, the efficiency might not be as high as what we're seeing here. We're seeing about 6% initial dose per gram of brain, which I think if we achieve half of that in the clinic, it would still be advantageous.
Thank you very much, Professor McGee.
Thank you for your talk. I just have a kind of a technical question.
Mm-hmm.
How do you do the correction for the fluorescent content in the brain? Because you have a Psi5, which is in the core of the micelle.
Mm-hmm.
So when you go into the endothelium, so you're going to be in endolysosomes. So when you consider the volume of the vesicle and the moisture content, so you are going to disrupt your liposome. It's not going to be at the CMC level. You're going to be well below the CMC level. So technically, your monomers are released. So the micelle giving you quenching, yet when you have the free chains, you get an enormous amount of fluorescence.
Sure.
So how do you do the correction in terms of what is really in the brain?
Yeah, the CMC we've not fully, but we have overcome the limitation of it by cross-linking the micelles with EDC, so creating a peptide bond between the cation and anion monomer. So we see very little disruption of the micellar structure following internalization. And we have shown that the micelles are stable in high sodium chloride solutions. So
have you looked into with endolysosomal enzymes, do you have any hydrolysis or not?
We have, yes, no, with the specific enzymatic metabolism of the micelles, we have not checked that yet. But yeah, that's a very good point, thank you.
Any other questions? If not, then this brings us to the end of this session. Thank you very much.